Good morning, everyone. My name is Harshit Kishore. I'm a student of B.Tech Computer Science from SRMU. My roll number is 054, and the topic of my presentation is glycolysis. So, starting with the topic, first we will see that what is the meaning of the word glycolysis. So, glycolysis comes from a merger of two Greek words. Basically, it is a Greek word. The one is glykis, which means sweet, and the other one is lysis, means breakdown or splitting. It is also known as Emden Meyerhof Parnas pathway or EMP pathway. So that was about the word. We'll move on with the introduction part. So, what is glycolysis? Glycolysis is the sequence of 10 enzyme catalyzed reaction that converts glucose into pyruvate with simultaneous production of an ATP. So in glycolysis, what happens? The glucose converts into pyruvate with the production of ATP. In this oxidative process, 1 mole of glucose is partially oxidized to 2 moles of pyruvate. Means 1 mole of glucose gives 2 moles of pyruvate. This major pathway of glucose metabolism occurs in the cytosol of all cells. This unique pathway occurs aerobically, means with oxygen, as well as anaerobically, means without oxygen also. It also includes formation of lactate from pyruvate. So we also get lactate from pyruvate, which is formed from the uh, conversion of glucose. And then the glycolytic sequence of reactions differ from species to species only in the mechanism of its regulation and in the subsequent metabolic fate of the pyruvate formed. In aerobic organisms, glycolysis is the prelude to citric acid cycle and etc. Glycolysis is the central pathway for glucose metabolism. So, glycolysis consists of two phases, namely preparatory phase, and the second one is payoff phase. So, glycolysis leads to the breakdown of 6C glucose into two molecules of 3C pyruvate, with the enzyme catalyzed reactions being bifurcated or categorized into two phases, which I have also told earlier, that is the two phases, preparatory phase and payoff phase. So moving further with the preparatory phase, what it is, it consists of the first 5 steps of the glycolysis, in which the glucose is enzymatically phosphorylated by ATP to yield fructose 1 and 6 biphosphate. Now this fructose 1 and 6 biphosphate is then split in half to yield two molecules of 3 carbon containing glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Thus, the first phase results in cleavage of the hexose chain. So, in the first preparatory phase, what happens? The cleavage in the hexose chain occurs. This cleavage requires an investment of two ATP molecules to activate the glucose mole and prepare it for its cleavage into 3 carbon compound. The last phase is payoff phase. This phase constitutes the last 5 reactions of glycolysis. This phase marks the release of ATP molecules during the conversion of glyceraldehyde 3 phosphate to 2 moles of pyruvate. Here, 4 moles of ADP are phosphorylated to ATP. Although 4 moles of ATP are formed, the net result is only 2 moles of ATP per mole of glucose oxidized, since 2 moles of ATP are utilized in phase 1. So, till now we have been seeing the summary of the two main phases of the glycolysis. Now we'll see the stepwise explanation of the glycolysis. So the first step is phosphorylation. What happens in this is glucose is phosphorylated by ATP to form sugar phosphate. This is an irreversible reaction and is catalyzed by hexokinase. So in this reaction, the hexokinase act as a catalyst. So we can see here how glucose is being converted into glucose 6 phosphate with the help of hexokinase, and the ATP is being converted into ADP. The next step is, that is step 2, is isomerization. In this, The rearrangement of chemical structure of carbonyl oxygen from C1 to C2 occurs, forming a ketose from the aldose. Thus, isomerization of the aldose glucose 6 phosphate gives the ketose fructose 6 phosphate. In the step 3, phosphorylation, what happens is here the fructose 6 phosphate is phosphorylated by ATP to fructose 1, 6 biphosphate. Now, this is an irreversible reaction, which means it cannot be reversed and is catalyzed by the phosphofructokinase enzyme. So, phosphofructokinase enzyme plays the role of catalyst in the step 3 of glycolysis, that is phosphorylation. Now, the next step, step 4, is breakdown. In this, the 6 carbon sugar is cleaved to produce two 3C molecules, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by aldolase. Now the step 5 is again isomerization. In this, the dihydroxyacetone phosphate is oxidized to form glyceraldehyde 3 phosphate. This reaction is catalyzed by triose phosphate isomerase enzyme. So this enzyme plays the role of catalyst in the fifth step, that is isomerization in the glycolysis. The next step is step 6. In this, two molecules of glyceraldehyde 3 phosphate are oxidized, and glyceraldehyde 3 phosphate dehydrogenase catalyzes the conversion of glyceraldehyde 3 phosphate into 1, 3 biphosphoglycerate. In the step 7, the transfer of high energy phosphate group that was generated earlier to ADP from ATP. This phosphorylation, that is addition of phosphate to ADP to give ATP, is termed as substrate level phosphorylation. Means the phosphorylation which occurred in which the addition of phosphate to ADP happened to give ATP is termed as substrate level phosphorylation, as the phosphate donor is the substrate 1, 3 biphosphoglycerate, that is 1, 3 BPG. The product of this reaction is two molecules of 3 phosphoglycerate. Means in the end of this reaction we get as a product two molecules of 3 phosphoglycerate. Now the step 8, what happens in step 8 of glycolysis is the remaining phosphate ester linkage in 3 phosphoglycerate is moved from carbon 3 to carbon 2 because of relatively low free energy of hydrolysis. Means in the step 8 the free energy of hydrolysis is relatively low, so the remaining phosphate linkage in the 3 phosphoglycerate is moved from carbon 3 to carbon 2 to form 2 phosphoglycerate, that is 2PG. Now the step 9, that is dehydration of 2PG. What happens in this is the second reaction in glycolysis where a high energy phosphate compound is formed. The 2PG, that is 2 phosphoglycerate, is dehydrated by the action of enolase to PEP, that is phosphoenol pyruvate. This compound is the phosphate ester of the enol tautomer of pyruvate, and this is a reversible reaction. So basically the step 9 of glycolysis is dehydration of 2PG, that is 2 phosphoglycerate, and this is the second reaction in where the high energy phosphate compound is formed, and the compound is phosphate ester of the enol tautomer of pyruvate. Now the 10th step is transfer of phosphate from PEP to ADP. This last step is irreversible transfer of high energy phosphoryl group from phosphoenol pyruvate to ADP. This reaction is catalyzed by pyruvate kinase. This is the 4 substrate level phosphorylation reaction in glycolysis, which yields ATP. This is a non-oxidative phosphorylation reaction. So that's all from my side. uh, I would like to end my presentation here. Thank you very much.